Hello everyone, hope you are doing great and welcome to the episode 3 of Python programming for biology, medicine and genetics. A very fantastic journey. I am Hamid Reza Bolhassani, PhD of computer engineering, researcher, data scientist and founder of DataBioX. I'm very happy to see you again in this episode and I hope that this episode would be useful and informative for you. So you remember in the previous episode we defined a problem of sequence data of an RNA sequence data, and we did something on formatting the data and some other operations using Python pandas library for doing these operations. Then in the previous episode we did the conversion from wide to long format. You see this data frame, we had a data frame named count. It was wide and we wanted to convert it to long format, so we changed this by the command by the methods that you remember already in the Google Colab. So in this episode we want to join two tables together. Joining two tables. You remember right now we have a sequence data, sec data data frame, and also a very small data frame named sample info with 12 rows and 4 columns representing our overall information about all the data. You see these are sample, and for better understanding you can again see this. This is the exactly, you see 3 category, each of 2 sample, 3, and totally these are 12. And as you remember this data, this is the sample info. Yes, so you remember we also changed one of the columns name. Now we have sample ID here, and also we have the name sample in the data frame sec data. So these 2 columns are common, both representing the sample. So it's very good here to join these 2 tables with a common column and just have one column named sample. So joining these will result in, for example, we will have a one table, all of this information, and it's very good because we see one sample here, each of gene, how many gene in the count. You see how many count of this gene for this sample, and also get characteristics. For example, here it's mammary gland, luminal cell and Virgin, and development stage and so on. So for visualization it will really help us, because without joining these 2 tables we cannot have we can't extract some meaningful information, some more meaningful information for discovery and for exploring what we need. Okay, so let's go hands-on experience on Python. So right now you see sec data and sample info. I remember that we define sample info like this. So I can because I want to make it like just this. Okay, because sample underline info is I think is more clear than simple info. You see this is the dataset, and also for getting more compact we will get ahead. For getting the 5 top rows of this data frame. Also we had count dot head, sec data sequence data dot head. So you see that the common things between these 2 tables is this sample ID and sample, and we wanna join them together. So right now we will introduce and will use the merge method in pandas. So pd.merge, and first, which data sets? Sec data. Yeah, we want to merge sec data here on left and sample info. Yeah, then we will say left on the left we have sec data. On the left we want to have sample, and right on in the right we have sample ID. Yeah, with the with capital S, sample ID. And the method how means alter. Okay, oh sorry, this should be ter. Yeah, we can see that we have this. Yeah, but there's the two tables are merged together, but we have two column named sample. We just need this, so we'll drop this. So let me put this in all info. Yeah, then I will get all info.head. Yeah, and then for dropping all info.drop, for drop this sample ID, all info.drop columns equal to and then we will use inplace true, because you want to replace it with the previous data frame with removing this column. Okay, so now we will get, yeah, that's right, very nice. So we can we will see that we have gene ID, gene symbol for each sample, count, and also the characteristics, immunophenotype, and also developmental stage. Yeah, and yeah, you see, for example, 2 days, like 2-day lactation. So one interesting thing that we can do here, we already joined these two tables. Yes, we want to check the, you see these are the rows and columns, totally we have the rows are same, and we have 7 columns. Yes, and we will get shape to check it, and you see all are the same. Yeah, so all info dot shape. Yeah, 284,008 to 820 and 7 column. That's right, and exactly like this. So we got everything here until now, and make a data frame containing all the data that we need. In the next episode we will go through the data visualization and some practice for visualizing the data here to extract meaningful information and storytelling, and something that help us to discover new things about this RNA sequence data. I hope that you enjoyed this episode and it was useful and informative for you. For any questions or any things that you want to share, feel free to contact me and share your comments. And if you liked it, kindly share it with your friends and colleagues who are interested. Hope to see you in the next videos. Bye.